Hi everyone, my name is Siripan Sungchoi. I am RJJ 23rd recipient and PhD student in Biodesign Medicine, Mahidol University, and my advisor is Assistant Professor Dr. Panat Anurakpida. Today I'm going to present about the first report of rhoptry protein gene of Babesia canis vogeli causing canine babesiosis. So, Babesia canis vogeli is a blood parasite in dog that transmitted by the Rhipizaphylla sanguineus or brown dog ticks. And in Thailand, the prevalence of the Babesia canis vogeli has been reported. And the clinical manifestation of this disease is fever and pale mucous membrane from anemia and also apathy and anorexia. In some cases, it possibly lead to death via the multiple organ failure. For the diagnosis method for the canine babesiosis that the most used at the present is the microscopic examinations because it's simple, not required any advances tool and also affordable price. But limitations of this method is require the expert for investigations and also come up with a low sensitivity and specificity. So the diagnostic test kit should be an alternative way because it can solve the problem of the sensitivity and specificity. However, the limitations of development of diagnostic test kit for detections of B. canis vogeli is there is no database or any information about the B. canis vogeli specific protein or genes. So in this research, we aim to study um, the specific protein gene of B. canis vogeli. I will wrap up a little bit about the rhoptry protein. It is the vital protein for the wholesale invasion process of the B. canis vogeli, for example, in tight junction step, in invasion step, or even the multiplications. For the material and methods. In this study, the blood sample will be obtained from the cooperative animal hospital or clinic, and then we do the DNA extractions and screen the positive sample. After that, we design the specific primer to detect the B. canis vogeli rhoptry protein gene. And when we already detect this gene, we do the gene clonings and extract the plasmid and then do the confirmations by gene sequencing. When we obtain the database of gene sequence, we do analysis in terms of phylogenetics analysis, protein characteristic, and also B cell epitopes. For the result of the detections of B. canis vogeli rhoptry protein genes in the positive sample, the agarose gel showed the target band at the 900 base pair, and we also confirmed by the sequencings. And for the phylogenetic analysis, we found that the gene sequence of the rhoptry proteins is conserved among the Thai strain and also conserved with the B. canis canis. However, we need to study more about the genetic diversity in other countries to more understand. And for the protein location and signal peptide prediction, we found that this protein is located outside the cell and also not contain a signal peptide sequence. And for the B cell epitopes, we found that this protein sequence contains 6 B-tail epitope positions that is important to be the antigen candidates for the diagnostic test kit developments. To sum up, in this work, we are the first report of the canine vogeli rhoptry proteins, and we also provide vital informations that might be influence the further study, especially in terms of developments of diagnostic test kit to detect Babesia canine vogeli. And I would like to say thank you to the other day PhD scholarships to provide the financial support to my study. And I would like to say thank you for attention, and any question are welcome.